My name is Travis Burrow with the IBS program. I'm here to describe translation and its processes to you. We're going to start simple definition, which is just the process in which you have proteins produced by way of mRNA coming in, coding for these proteins, and then the ribosomes are the ones that decode these mRNAs and help the proteins to be assembled. And so the process starts, let's talk about the process as a whole. It has three parts to it. It starts with initiation, goes into elongation, and then it ends in termination. Initiation is the process by which this mRNA you see here actually binds to the ribosome. We have elongation, which is the process by which these peptides are added to this growing amino acid chain. And then you have termination, which is the process where you have a stop codon coming into this active site and stopping the peptides from being added, and it then disassociates from the ribosome, ending the chain of protein being produced. And so the ingredients required in order to perform this process, you need to have mRNA, as we mentioned, which serves to bring in the template for your protein. You need your transfer RNA, which serves to bring in the amino acids to add to the growing peptide chain. You need ribosomes, which serve as the site for this process to occur. And then, of course, you need nucleotides that are matching on the anticodon to produce these peptides. All right. And so the initiation of the process starts, as we mentioned earlier, with the mRNA binding to the active site here. You have a start codon, which is methionine, which is coded AUG. It starts at the 5' end and is read all the way to our 3' end. And so this mRNA, in addition to our ribosome, once they bind, this creates what's known as our initiation complex. And then next we have our elongation occur. Elongation, as we mentioned in a nutshell, is where you have these peptides being added to this growing chain. The way this works is each one of these, there are 3 codons in mRNA that code for a specific amino acid. These have matching anticodons that are presented in the tRNA. And as this binds to the active site here, the A you see, have this tRNA come in and have the corresponding matching anticodons. And from that, you then have an association from this peptide that's attached to our tRNA, attached at our P site, and it starts to grow our peptide chain. And so then if it's our first one, you would have, like if we say our start codon would be methionine, that would then move into the E site for exit, and you would have the next one come up. And so in termination, let me go back. I got a little ahead of myself. For termination, it's where you have a stop codon, as we mentioned, coming to this A site. And once it gets into this A site, that's when you have a hydrolysis occur between this peptide and the anticodon, and so then this would then disassociate, and you would have a free floating then polypeptide within your cytoplasm. And so that's kind of translation in a nutshell. But again, you have initiation, you have elongation occur, where you have the growing peptides added by way of your tRNA. Then you have termination occur. We have the stop codon coming to one of 3 stop codons coming to this E site, A site. I'm sorry, and that initiates hydrolysis, and again, disassociation between those two. And so once you have translation occur, a lot of times we found that there are many proteins, specifically sex hormones, tend to undergo some of these post-translational modifications in order for them to be active. And so what happens, and those are a number of cellular processes. You can have methylation occur, where Typically, this deactivates or silences a lot of genes. Excuse me. You can have hydroxylation occur where there's a hydroxyl group added to your gene. There's an acetylation that may occur. Phosphorylation is another process that may occur where you have a phosphate group added, which tends to activate enzymes a lot of times. We're also prone to see what's known as glycosylation, where you have a glucose added to it, a sugar moiety added to it, which changes it to some effect. You also have adenylation as well as ubiquitination, where you have ubiquinone maybe added to it. But all of these change the way that protein may function or interact with other proteins, as well as other organelles within the cell. And so a protein that's undergone these post-translational modifications will be different from one that has not. And so now we'll talk about some of the features of these three processes and how they kind of overlap and intertwine with one another. One thing that is important to point out, all of them have the same steps, which starts with initiation, of course, and you have elongation as well as termination, summing up the processes. The directionality is going to be the same in all of them, where you have 5' going, you have the process going from the 5' direction to the 3' direction. The only other time that you would see it go in the opposite direction would be when you have DNA replication, where you also have some 3 to 5' action occurring. It's also important to remember that the atmospheric conditions, like pressure, various concentrations of solutes, temperature, all those things are going to be the same to make sure that that protein is formed properly. And so those are all going to be the same because in the cell, pretty much going to be the same requirements to keep this cell alive, or functioning rather, at the ribosome, the same as it's going to occur in the nucleus. So it's important to keep all those things static. You do see some differences occur as far as location when these processes happen. Replication, where you have the DNA being copied, basically occurs in our nucleus, where you see transcription and translation tend to occur in our cytoplasm. You have different cellular products produced. When you look at, replication, the end goal is to have new daughter cells that form from these new DNA strands that are formed. When you're talking about transcription, the ultimate goal is you're trying to produce an RNA molecule from which a protein template can be produced. And so in translation, which is we just talked about now, the ultimate goal is to have a protein that is produced. And so another difference is important to note is the amount of genetic material that's being decoded. When we talk about replication, because we're producing two new organisms, we're producing a new organism, two new DNA strands rather, you have the entire genome being replicated. When we're talking about transcription, a lot of times those are just portions of the DNA relevant to a particular processes or circumstance. And we'll talk about translation. Translation just tends to be genes that are decoded at a time. Okay. And so we'll look now, a couple more differences. We have replication and translation. Those tend to start at one particular point of origin on the DNA strand, on the genetic template, as opposed to transcription, you will have occur at multiple points on the DNA. Another difference we'll take a moment to point out, when you're looking at replication and transcription, those tend to start with the DNA template, whereas translation, you're producing your protein, you're going to start from RNA. And so now I'll touch kind of briefly on the enzymes necessary when we look at a replication. You're looking at a DNA-dependent polymerase, you're looking at helicase, primase, ligase, topoisomerase 1, gyrase, which is topoisomerase 2. You need these DNA binding proteins to prevent the DNA from kind of coming back together. And then you also need telomerase, where you're looking at eukaryotic cells. When we're talking about transcription, you're going to have an RNA-dependent polymerase, and pretty much most of those other enzymes except for telomerase. So you'll still need helicase, you'll still need a primase, ligase, topoisomerase 1, as well as topoisomerase 2 or gyrase. When we look at translation, that list drops as far as numbers are concerned, and we're looking now at an RNA-dependent polymerase, mRNA, rRNA for a ribosome, and tRNA. And so another couple of differences. One, when we look at fidelity, as far as maintaining the same, making sure there aren't any errors. Okay, which we'll have my colleague talk about here in just a moment. When we're looking at errors, transcription and replication, there are very few errors. Things are pretty much copied exactly as they're seen. However, once you get to translation, the fidelity decreases, and there's a concept known as the degeneracy of the genetic code, in which you have basically the last amino acid that's coded for can have a number of different codons code for it. And so the first of the three codons, the first one is going to have the most fidelity. The second one is going to have decreasing fidelity, but the last one is almost obsolete as to which one is used, because it's going to still give you that same protein. And that ensures that you don't have, well, I'll let my colleague talk about that one. But we have also, I guess lastly but not leastly, we'll talk about the nucleotides that are used in replication. You're going to have adenine, thymine, guanine, and cytosine. But when you're taking a look at transcription and translation, you're going to be utilizing uracil in place of thymine. And so that is my brief lecture on translation. And thank you for listening. Hopefully that was helpful.
I am Bulbul Ahmed, PhD student here at Tuskegee University. Today I am with my colleagues, friends. We are discussing on central dogma. Previously, Travis Barrel had explained the post-transcriptional method, and now I am describing the mutation in the central dogma. So first I have to discuss that what is mutation. Mutation, I think that mutation is the main factor by which the variation of an organism is happened. In scientifically, we can describe it that mutation refers to the deletion, duplication, rearrangement of the larger genes of the or insertion in the nucleotides. We know that in DNA there is the nucleotides combination A, T, C, Z are distributed in A, G, U, or the nucleotides are distributed like that. A. That means 3 allele makes a codon. A, C, G. This type of 3 nucleotides are making the codon, and mutation is happening and mutation is the combination of these nucleotides. So if any deletion, duplication, rearrangement happened in the nucleotides combination, then it is called mutation. And it is very important for the living organism to make the variation of organism to organism. Suppose you think when human, if the human's nucleotide is not rearranged, is not mutation is happened, the all human will be the same color, will be the same height, will be the same structure. But due to the mutation only, the variation is happened. So, okay, now we understand that what is mutation, and now I want to explain the 3 things. That means deletion, insertion before that I want to discuss that through mutation okay, through mutation what has happened? New alleles generation has happened, variation occurred, mutation creates variation in protein coding portion of genes that can affect protein itself. And how the mutation is happened in the organism due to radiation, chemical effect, byproduct of the cellular metabolism, free radicals effect, ultraviolet ray of sun. These agents can cause the mutation in organism. Most of the time mutation is reversed and DNA repairs contain, and also DNA repairs contains happened by the DNA. So now I want to show some mutation. Okay, if I see that the codon of a DNA is AAA, ATZ, CTT, CTC. This is a normal DNA, and if we then mRNA we can get ZAA and ZTA, C. It is what? This is phenyl. This is tyrosine. This is glutamine, and this is this is also glutamine. And when the substitution is happened, when the substitution, that means the change of the when substitution is happened, here we see that the this is uracil, UAC, the G turns into A, and ZAC. Here you see the lysine. Lysine has come. Just substitution. We are changing the G into A. And another one, if I say the insertion. If the insertion is happened, suppose here is I want to insert a C, then it will be just here is C, C, T, T, C, T, C. Here just if we add a C, then it become a new glycine. So it is insertion. And second, and also if you delete a nucleotide, then if you deletion is happened, suppose this we want to delete, A is deleted, then T, G, C, T, T, C. So here the new is coming tyrosine, TSR, tyrosine has happened. So here we are seeing that due to substitution, insertion, deletion, the nucleotide substitution, insertion, deletion, the nucleotides is changing, new the protein code, protein is forming, and as a result the mutation is, with this way the mutation is happened. And another type of mutation I want to discuss here, that there is some silent mutation happened. There is, if the after deletion, substitution, or any kind of the silent mutation is a point mutation, wherein the codon codes, the changing codon codes will not affect the protein product. This is called silent mutation. Suppose TTC is a lysine, and if we change the C, then T, this is also lysine. So if the deletion, after the deletion or duplication, if the protein is not changed, then it is called silent mutation. And there is some other point mutation we want to say. The other one is nonsense mutation. That means ATC. If the A is turned into UAZ, then this is stop codon UAZ. So this is called nonsense mutation. And missense mutation, TCC, if the mRNA we can get AZZ. This is arginine. This is just conversion. This is just a T turned into A, C, G, C, G, AZG, ARG. And another is frame shift mutation. In this frame shift mutation, the deletion, in this frame shift mutation, we can see the adding, deletion, or any type of deletion or insertion of a nucleotide, but nucleotide, and it will be it differs that in insertion or deletion of nucleotide base in numbers are not multiple by 3. If we see C, T, A, G, C, here if we delete, then C, T, A is deleted. A is deleted, then G, C, T, and if this is not divided by 3, then it is frame shifted. That means frame changes, and insertion also be happened. Okay, insertion also be happened. C T A here if I inside the here if I add a Z, C T A Z A or another things happen. So that is the new is G is added here. So deletion and adding may be happened, but the nucleotides are not divided by 3. Then the frame, it is called frame shift mutation. And other types of mutation also we can show that there is some inversion also. Suppose the original this is a original is T A C T Z A Z Z. If the inversion is changes, that means A become T or T become A or T become A. That means inversion. Inversion happened, then it is called the interchange the place. Then it is called inversion mutation. And so there is some transition inversion. When the mutation is responsible to change the purine to pyrimidine or pyrimidine to purine, then it is called transition. And transversion also happened when purine to pyrimidine is occurred. Translocation also happened when A come to T, T come to A. This is the translocation. And then is the duplication of a nucleotides is happened. Suppose T A or C, the duplication is happened. Then it is called the tandem. So this is about the mutation in the central dogma. I think you all enjoyed it. Okay, bye. Thank you.